Okay, so I think that clock is just a few minutes fast, but we'll talk about the course. So you have in your possession the syllabus, and the syllabus is very similar to the one that you got in 231. So the proportion of assignments and grades are about the same. If you go on Canvas, you'll notice that there is a homework that is due on Sunday, next Sunday at midnight. And then 2 weeks from now, you're going to have a PhysioX that is due. So you want to make sure that you finish that because that is worth 36 points. So the PhysioX is going to be over neurophysiology, which is actually what we're going to be talking about today in class. So hopefully it'll be a good review. We'll have our first quiz next Monday. And it'll be 15 points, and it'll just be over the information presented today, and then Wednesday, and we'll have lab this week as well. So are there any questions about the course? It just depends upon how things fall. Like if we miss a class, then that goes to Wednesday. So generally, if I don't miss any classes, then it doesn't change until after Memorial Day because we have a Monday off. And then the quiz is on Wednesday. Any other questions about that? Okay, so we are going to start today talking about the nervous system. And this is going to move into a discussion also about the skeletal muscles, because you'll notice here when we look at this outline of the nervous system, we have sensory information that comes in to our central nervous system, and there it is integrated. And then from there, we have neurons that go out and then innervate the skeletal muscle. So today we're going to, the first part of the quarter deals with the nervous system, and then we move into muscles. So I'm doing it a little bit backwards from the order in which they're presented in the book. I believe the muscles are presented first. Okay, so when we look at the nervous system, this is composed of cells called neurons. And neurons have a very special characteristic in that they are able to receive information and then transmit information. So they receive and transmit information. So when we talk about the functional aspect of this, we can talk about sensory neurons, right? And our sensory neurons are able to either connect to our, in this case, for example, to the neurons that are in our eyes, which are called photoreceptors, that absorb light energy and then transmit it into the central nervous system. And so if you remember back when we were talking about homeostasis, and we talked about the different pathways. And so sometimes sensory neurons are referred to as afferent neurons because this is the information that is coming in, that is being received. So as in like homeostasis, where we have a receptor and then the receptor sends it to the central nervous system, that's the afferent pathway, okay? And then we have neurons that connect sensory information with motor output. And these are what are referred to as interneurons, right? So in between. So these could be found in the brain, but they can also be found in the spinal cord. So for example, with the knee-jerk reflex, when we stimulate the muscles, it actually sends a signal to the spinal cord, which then causes your knee to jerk. And all of that integration occurs in the spinal cord rather than in the brain. Okay, so we have interneurons. And then we have what are called motor neurons. And motor neurons connect to muscle. So these are connect to muscles and glands, for example, and even the heart. Talk about motor neurons. And these are called efferent because they cause the response, right? So So the efferent pathway of the homeostasis pathway causes the response to the stimulus. Okay. So we're going to look at more about what these neurons look like and the structure of them. But first, what I want to do is I want to kind of outline the nervous system. So in the front half of your book, or in this first chapter, they outline the different parts of the nervous system, and this will kind of give us a perspective as to how the neurons are connected to one another. Okay, so we have the central nervous system. So this is called the CNS. Probably should close my doors. I have this new screen, so it's a little bit, I'm trying to learn how to use it. Okay, so this is the central nervous system. And we'll call this the CNS. And this sends a signal to and from the peripheral nervous system, which is called the PNS. So this is my PNS, okay? So from the peripheral nervous system, we have a branch out here, and I'm just gonna draw the arrow going in one direction because this is sensory. So sensory information travels into the peripheral nervous system, and then it can be integrated by our central nervous system, okay? And then coming out of the peripheral nervous system, we have what is referred to as the motor. And then from the motor, we can send information out to the somatic division. So this is called the somatic division. And so what does soma mean? Does anybody remember? Does anybody know what soma means? Nobody knows what soma means? Soma means body, okay? So this is skeletal muscles. Okay. We also have autonomic division. And we kind of looked at the autonomic division when we were talking about our diving reflex last quarter. So what are the two parts of the autonomic nervous system? Yes, so we have the sympathetic and we have the parasympathetic. And I'll just abbreviate that, okay? Now, remember that the sympathetic is sometimes referred to as the fight or flight, and we'll talk more about its characteristics. And the parasympathetic is considered relaxation and conservation of energy. Now, there is another part to our nervous system, which is kind of interesting, and I'm just gonna draw it up here. And I'm going to say, this is my enteric. Does anybody know what the enteric nervous system does? So there's been a lot of talk these days about like your microbiome and how the microorganisms in your digestive tract can affect your mood, for example, can also affect your body weight. So the types of microorganisms in your digestive tract can sometimes speed up metabolism or decrease metabolism. And so when we talk about enteric is in your gastrovascular system. So that is in the digestive system. So you can write digestive system under enteric. And sometimes scientists are kind of now referring to that as your second brain. And the really interesting thing about the enteric nervous system, which is all the nerves and the nervous tissue that's in your gut, is that it is seems to be able to run completely on its own without any input from the central nervous system. So that's why it's sometimes referred to as the second brain. And we'll talk more about that when we start talking about that part of the system, okay? So this is the outline of your nervous system. Okay, so we're gonna talk about the nervous tissue. And nervous tissue consists of neurons, but it also consists of what are called glial cells. Sometimes these are called neuroglial or neural glia, okay? So the glial cells are actually, and when I was studying this, we didn't even talk about these. So these are actually kind of recently discovered and also kind of recently realized how important they are. So these are the helper cells. So when we look at the glial cells, we can talk about those that are in the central nervous system. Okay. And so one of these is what is referred to as an astrocyte. So what does astro mean? What is astrology? The study of the stars, right? So these are star-shaped cells, and these are really important for regulating the external environment of the neurons. So these regulate the external environment of neurons. And this is important because the neurons are so specialized that they kind of have to be taken care of. And so stuff that diffuses into your brain, for example, via capillaries, these astrocytes make sure that that stuff that diffuses in is of the proper concentration and that it's not any harmful stuff to your neurons. So those are called the astrocytes. Okay, we also have cells which are called microglial cells. And these are actually, it's kind of interesting, they're called micro because they're smaller, but they're actually similar to macrophages. And so these actually have an immune function. So they think that these cells clean up any debris, and they actually can also go in and remove synapses. So we have what is called neurological pruning. So when we are born, we're actually born with many more connections in our brains. And then what happens is that the cells start to die and connections start to be broken. And only those connections that are stimulated by the environment remain. And so the microglial cells not only clean up the environment and remove old dead cells, but they also help to create neurological pruning where they decrease the number of synapses in the brain. And so we're going to talk about Alzheimer's, but Alzheimer's is an example of a neurodegenerative disease. So we can talk about neurodegenerative diseases. Are primarily caused by inflammation in the brain. I'll just put influenced by inflammation in the brain. Okay, and so it's the inflammation. These microglial cells are good, but they also can lead to chronic inflammation, which is a bad thing. Okay, so that can cause nerve cell death, right? And then that could cause the brain to start to shrink. And those are two signs of diseases like Alzheimer's disease and Parkinson's disease. Okay. We also have cells which are called oligodendrocytes. And you might have seen the dendrocyte before. Oligo means few, and dendro means branching. So these cells have few branches, and they wrap themselves around the axon. And we'll talk about what the axon is. And the axon is found in the neurons. And what they do is they help to speed up transmission of the signal that the neuron is sending. So those are the oligodendrocytes. And then we have ependymal cells. And this is the last type of neuroglial cell in the central nervous system. And the ependymal cells have cilia. And they are associated with capillaries around where the cerebral spinal fluid is produced. So cerebrospinal fluid, when we think about our nervous system, it is hollow. So even our brain has ventricles in it, which are spaces that are filled with fluid. And our spinal cord also has a central canal that is filled with fluid. So the cerebrospinal fluid is abbreviated as CSF. Okay, so that's the cerebrospinal fluid. Okay, so you need to be able to tell me the function, but you also need to be able to identify them. So in your book, there are pictures of the different oligodendrocytes. Oops. Oh, shoot. Too sensitive. There we go. That is a good question. Look in your syllabus. What chapter are we on? 11. Chapter 11, okay. Okay, so these are the astrocytes. Notice that they have many branches. They look like stars, right? Notice that they are kind of wrapping themselves around the capillaries so that anything that comes out of the capillary first has to be monitored by the astrocyte, okay? Here's the microglial cells, right? So these are part of the immune system. And then when we look at the oligodendrocytes and the ependymal cells, this is the oligodendrocyte, and this is the area where the cerebrospinal fluid is produced. Cerebrospinal fluid is continually produced, circulated, and reabsorbed. So this is the fluid-filled cavity, like a ventricle in the brain, or the fluid-filled central canal of the spinal cord. And then this is the oligodendrocytes. Now, the oligodendrocytes create the myelin sheath. And you might have, if you've taken psychology, you might have heard about the myelin sheath being produced by a different type of cell, because this is what produces it in the PNS, okay? So in the peripheral nervous system, we have cells that are called Schwann cells, right? So they would be homologous. They would have the same, or analogous. They would be the same types of cells, but as the oligodendrocytes, okay? So the Schwann cells wrap themselves around the axon and they create the myelin sheath. So they create the myelin sheath around the axons. That says around the axons. This is myelin. I guess I have to work on using this pen. Okay. Then we have one more. So we have what are called satellite cells. And these are found in where the cell bodies are in the peripheral nervous system, which are called ganglia. But they might have the same function as the astrocytes. And so they just kind of help to create the proper environment. So the Schwann cells and the satellite cells are only found in the peripheral nervous system. Okay. And then the ganglia that are also in the peripheral nervous system. Okay, so this just shows a satellite cell, and in this case, it's called a satellite cell, because it's around the soma, the cell body, right? So like, our moons that orbit the Earth, that's what the satellite cells look like. Okay, so let's look at the neurons. So we have the cell body. Right. This is also referred to as the soma. So where in the cell body do we have the genetic material? Where is the genetic material found in the cell body? Nucleus. Okay. So this is what contains the nucleus. Sometimes I think it's not just me, it's like, okay, there we go. Nucleus. Okay. We also have cells that are organelles that are in here that produce energy. So what part of the cell body produces energy? Mitochondria. So they have the mitochondria there. Oh. Mitochondria. Okay. We also have the part of the organelle, the cell body, that produces neurotransmitters. And neurotransmitters are released to the outside of the cell. So neurotransmitters can be like proteins that are produced and then secreted. And so where are proteins that are produced and secreted, where are they produced in our cell body? Does anybody remember? The rough ER, I think I maybe heard somebody say this. So remember that the rough ER stands for endoplasmic reticulum. This produces the neurotransmitters. Right? So the neurotransmitters are produced in the cell body, but they're released out by the axon. And so we're going to talk a little bit about how they are transported in a minute. Okay. So when we look at the cells of the cell body and the neurons are long-lived, right? And they are largely amitotic. So that means that they do not reproduce. So A means without mitosis. So this is a big problem when we have brain damage or when we have like a severing of the spinal cord, is how do we get those cells to divide and reproduce and produce normal, healthy nervous tissue so that we can repair it, right? And so there's lots of interest in how to get or how to stimulate neurons to reproduce so that we can repair damaged tissue. Okay. We also can see that they have extensions, cytoplasmic extensions. And the receptive extensions, the ones that receive information, receive and bind neurotransmitters, are called dendrites. So dendrites have on them receptors that bind neurotransmitters. Oops, let me erase that. Okay, so they have receptors that bind the neurotransmitters. And so they are said to be the receptive surface. Right? So dendrite means tree. So sometimes these dendrites are highly branched. So there's lots of them. In most of our neurons, there's lots of dendrites that extend out, and they actually connect to thousands of other neurons. Okay. So the other cytoplasmic extension is the axon, and there's only a single. So this is a single extension arising from a part of the cell body called the axon hillock. Okay. So this is kind of a hill, right? But it's just kind of an enlargement of the cell body. Sometimes when you look at these cells under the microscope, you can't tell the difference between the dendrites and the axons. Okay, so sometimes it's difficult to tell. These have axonal terminals at their end, and so sometimes they're branched. And the axonal terminals function to produce and release. So they don't produce, but they release neurotransmitters into the synapse. And the synapse is just the space between neurons. And the space is really, really tiny. It's really, really narrow. But these neurotransmitters have to diffuse across that space and then bind to the dendrites on the next neuron, which is sometimes referred to as the postsynaptic neuron. So if we look at a diagram, you should be able to, not sure where that went. Down here? No, down here. Okay. So if you look at the next slide, okay. So you need to be able to identify on this diagram the different parts, okay? So here we have the cell body, right? These missile bodies are actually the RepR, and so they kind of stain darkly. So that's the RepR. You can see there's a mitochondria in there. So neurons use a lot of energy. They need a lot of oxygen and they need a lot of glucose. So these are the receptive regions, the dendrites. This is the axon hillock, right? And then this is the one axon that comes down, and these would be the axon terminals. And so you can see here that this particular instance, this is a peripheral nervous system, a neuron in the PNS because it has Schwann cells, right? And so you notice that the Schwann cells have actually wrapped themselves around, and we'll talk about the importance of that, but it helps to speed up the transmission of the impulse down the length of the axon. Okay, so when we look at Alzheimer's disease, there's a couple of characteristics. So if they take a biopsy of a person's brain who has died of Alzheimer's, this is what they commonly find. And so outside the cells between the neurons, they find what are called amyloid plaques. I might try writing with my finger. That might work better. Oops, plaque. So it's a QES, amyloid plaques, okay? So these are proteins that clump, and these are found between neurons. So this is clumps of proteins. The other thing that they find is something that is actually inside the neurons, and these are called neurofibrillary tangles. So these are in the neuron, and they're caused by defective tau proteins. So this protein is part of the system of kind of like railroad tracks that transport neurotransmitters from the cell body out to the ends of the axons. And so these are associated with microtubules. So associated with microtubules, and that's part of the cytoskeleton, if you remember from cell biology. And the microtubules are like, that's how things are transported. And I'm going to show you a video that shows that transportation of like neurotransmitters out from the cell body out to the ends of the axons. Okay. Tau, T-A-U, tau. Okay, so what happens when you get this is that the neurons start to die, right? So the neurons are not able to receive proper information, and they sometimes become deformed, and maybe even the microglials go in and cause inflammation in the brain, and they might even start attacking the defective neurons. And so when you look at a biopsy of a person's, look at the brain of a person with Alzheimer's, what seems to happen is that the brain shrinks, right? And so it also develops holes, and that's especially a problem in parts of the brain associated with the hippocampus, which is associated with memory, right? So then people start to lose the ability to remember people and things in their life, like what they just said, where, you know, who the people are, even close relatives. Okay. So we're going to watch a little video, just a short little video that talks about the normal, healthy brain and nervous system, and then what happens with Alzheimer's.
The brain is a remarkable
Oh no, I didn't check the music. Okay, so how do I get it to go through the speakers? Let me see if I can figure that out. PC, I don't want it to go through there. So over Christmas break, they did lots of things to change what's going on here. So let's see. Computer. You know, we might just have to listen to it through this today, and then I'll figure it out. Okay, we'll figure this out. Okay, so it's up as high as it goes.
Complex
Can you guys even hear that at all?
processes take place within our brains that let us speak,
You can?
move, see, remember, feel emotions, and make decisions. Inside a normal, healthy brain, billions of cells called neurons constantly communicate with one another. They receive messages from each other as electrical charges travel down the axon to the end of the neuron. The electrical charges release chemical messengers called neurotransmitters. The transmitters move across microscopic gaps or synapses between neurons. They bind to receptor sites on the dendrites of the next neuron. This cellular circuitry enables communication within the brain. Healthy neurotransmission is important for the brain to function well. Alzheimer's disease disrupts this intricate interplay. By compromising the ability of neurons to communicate with one another, the disease over time destroys memory and thinking skills. Scientific research has revealed some of the brain changes that take place in Alzheimer's disease. Abnormal structures called beta amyloid plaques and neurofibrillary tangles are classic biological hallmarks of the disease.
Oh, what did I just do?
Plaques form when specific proteins in the neuron's cell membrane are processed differently. Normally, an enzyme called alpha secretase snips out a precursor protein, or ATP, releasing a fragment.
Okay,
A second
we'll stop that.
gamma secretase.
Okay, I don't know what button I just turned the power on for everything, and then just let's see. Okay, hopefully I can get that back. Okay, I'll post that on Canvas in case you're interested. But it talks specifically about the role of the microtubules in transporting the neurotransmitters down the length of the axon. Okay, so we talked about how we have sensory neurons and motor neurons and interneurons, but there's also a classification based upon structure. So we talked about function, right? So that would be sensory, motor, and interneurons. But we also have structural. Okay. So function, sensory, motor, and interneurons. Okay. So structural includes multipolar. So what this means is that they have many branches, and particularly more than two. So they have 3 or more branches, and this is the most common, right? So 3 or more extensions. So most of the neurons in our body are multipolar, and I'll show you a diagram of this in a second. But we also have some neurons that are called bipolar. And this means that they just have one dendrite and one axon. And so we're going to talk about the neurons that are in your retina of your eye. And so an example of those would be bipolar. And so the neurons that are in your eye, specifically the retina, the nervous tissue in your eye, are bipolar. And then we have ones that are called unipolar. And this means that the axon extends directly or just from, so there's just one axon, one axon extends from the cell body. And so the way that it looks is that we have a cell body out here, and then the axon extends here in this direction and in this direction. And then on one end, we have the dendrites. And so that's the unipolar one. And this is very common for sensory neurons coming into the spinal cord. Okay, so sensory neurons coming into the spinal cord are said to be unipolar. So in your book, they have these nice pictures of the different types and tables and diagrams, right? So here would be multipolar, right? So we have, just like in our diagram, we have many dendrites, one axon. Here we have, from the cell body, only one dendrite and one axon. So that would be bipolar. And then in this one, the cell body is right in the middle of the axon. And in one part of that, one end of that, that's the receptive endings. And the others are where the neurotransmitters are released. Okay, so when we talk about the electricity in the brain, right? When we talk about electricity, electricity is just a form of energy. Okay, and so it is the movement of charged particles. Now, in our electrical systems, like in this building, the charged particles are electrons, right? So the electrons flow along electrical lines. But in our bodies, we don't use electrons, but rather we used charged ions, including sodium ions. So that would be Na, right? Potassium ions. Okay, so that's potassium, sodium, potassium, and chloride ions. Okay, so I'm going to put not electrons. Okay. So the way that these ions move through the plasma membrane is through the use of channels. And channels, you remember when we talked about the plasma membrane, are proteins. So we have proteins that are embedded in the phospholipid bilayer, and that is what allows stuff to come into the cell and out of the cell. So we can talk about passive channels, or what are referred to as leakage channels. Okay, so these are just open all the time. So that's why they're called leakage, is that potassium and sodium and chloride can come into the cell and then they can exit the cell, right? So they're open at all the times. And so we say that they are not gated. So they're not, you can't close them and you can't open them. They're just open all the time, okay? Now we can also look at gated channels. And some of these gated channels are chemical. So they have chemically gated channels. And what this means is that if something binds to the protein, it will open, or sometimes it can cause it to close. But generally what it means is that if a chemical binds, so if a neurotransmitter binds, the channel opens. Okay. We also have what are called voltage-gated channels. Oops, not gated, gated channels. Okay. So we're going to talk about voltage in a minute, but what this means is that a difference in charge causes the channel to open. So a change in charge. Okay. And so these movement of ions into and out of the cell is actually what causes the membrane of the neuron to change and actually is able to send a signal or an impulse. Okay, so in your book, they have pictures of these different channels. So this is one, this would be a chemically gated one right here, because this is the receptor. So the neurotransmitter can bind to that receptor, and then all of a sudden the channel opens up. Okay, if it's closed, the sodium is not able to move in, but if there's a change in charge, then the gate opens and the sodium can flow through. Okay, so this just shows the difference between chemically and voltage gated channels. Okay, so let's look at what this has to do with neurophysiology. Okay, so when we look at what is called the resting membrane potential, we can define membrane potential is equal to the difference in charge between the outside of the neuron and the inside of the neuron. When we look at the resting membrane potential, this is actually measured in millivolts. So we'll put measured in millivolts. Okay. So if I looked at my axon, for example, so this is my axon, and this is outside, and this is inside. So this is my axon. What we see, if we put a voltmeter down in here, is that the inside is negatively charged compared to the outside, which has a positive charge, right? And so if we measure the difference between those two, it's about a negative 70 millivolts. So MV is millivolts, okay? So that's the resting potential. So a neuron that is not firing, and you saw it just briefly in that video, the firing is like a light that comes on and moves down the length of the neuron. If it is not firing, it is at a negative 70 compared to the inside compared to the outside. So it's just a difference in charge. And they were actually able to first discover this by using giant neurons that are found in squid and octopus. Those guys have these giant neurons that you're actually able to manipulate fairly easily. And oh, I thought I had a picture of it, sorry. And manipulate fairly easily to allow you to put a little voltmeter down inside this to measure the difference. Okay. Okay, so let's look and talk about what causes this difference in the charge. And so we need to talk about the difference in ion concentrations. So I'm going to draw my axon again. Okay. And I'm going to draw sodium ions as being really concentrated outside and not so concentrated inside. I'm going to draw potassium ions as being very concentrated inside, but not so concentrated outside. So that should be a smaller K. Okay. And then we have what are called anions. And anions are negatively charged proteins, right? And they are only in the inside, right? So these differences in the positive charge are important, but it is actually the relative concentration of the anions that actually makes the inside negative when it's at rest. Okay. So this neuron has leakage channels. So which way would sodium move? Which way does sodium diffuse? Would it diffuse in or out? What did they say? In. Okay. So sodium is going to leak in to the inside of the, okay, and potassium is going to leak out. Right? So this is leakage. I'll put leakage. And potassium actually leaks out faster than sodium leaks in. And so that is also the reason why the inside at rest is negative. So the potassium, the membrane is much more permeable to potassium. So this leaks out faster than the sodium would leak in. Now, in order to maintain the difference, what would happen over time is that you would get equilibrium. So you predict if you didn't do anything, put any energy into the system, that over time, the sodium and the potassium should be equally distributed outside the neuron as opposed to inside the neuron, right? So there would be equal concentrations. And so what keeps the concentrations different is what is called the sodium-potassium pump. So when you think of a pump, you think that it takes energy. So this takes ATP. Okay. And as the sodium moves in, right, it is pumped out. So sodium pumped out against its concentration gradient. Right? And potassium is pumped in. Against the concentration gradient. Okay? So diffusion is passive. It takes no energy, but to pump something against its concentration gradient is like pumping water up a hill. Right? It takes energy to do that. And the whole reason that we pump them is that we need to maintain these differences in concentration so that when the neuron gets stimulated, it can open up gated channels and cause the ions to move in such a fashion that the charge on the inside of the neuron will change. Okay. Is everybody following me so far? Okay. So this is at rest, right? So I'm going to put resting neurons are energetically expensive to maintain. Right? So there is a kind of a trade-off between having a big brain and then maybe losing part of that brain, right? So if we have a big brain, then we are going to be using a lot of energy to maintain. Even the neurons at rest take energy. And so there's been a lot of study about the plasticity of brains in vertebrates and even in humans. And so one really interesting thing that they discovered is that in some birds, like chickadees, and chickadees overwinter here. So those are those tiny little birds that have like they're white and have a black chin and they go chickadee, right? So you hear them in the wintertime. They actually hide their food in caches around their neighborhood. And in the winter, they go to those caches and they have to remember where they are, so where they've stored all of their food. And so their brain, that area of their brain that remembers that, expands during the winter months. And the really cool thing about the chickadee is that in the summertime, when it doesn't have to remember where it has cached its food, that part of its brain actually shrinks. So neurons die and neurons are produced. And that is huge news for people that are studying the nervous system, because that means that those neurons are actually reproducing, right? I mean, that means that they can replace themselves under certain circumstances. And so this has relevance for humans, because what they've discovered is that people that are under intense stress, their hippocampus, the part of their brain for long-term memory, actually shrinks. So if you're really stressed out, your hippocampus is actually going to shrink a little bit, and that actually conserves energy. And then when you're not stressed out, your hippocampus can get bigger, right? So being stressed out is not a good idea for retaining information, right? And so the plasticity of the brain seems to be greater than what once was thought, right? So our brain might shrink when we're under stress, and it might expand when we're not. Okay, so in your book, right, they show the concentrations of the sodium and the potassium. Here, potassium is being pumped in, and for every 2 potassium that is pumped in, there's 3 sodiums that are pumped out. So this also shows that there is more potassium ion channels in the membrane. So potassium is going to leak out faster than sodium is going to leak in. So this also creates the negative charge. But we also have these anions, right, in there that also create the negative charge. And so potassium comes back in, then that puts us about the resting membrane potential, which is a -70. So if potassium wasn't leaking in, the membrane potential would be even higher. It would be like a -90. Okay, so let's talk about what an action potential is. So an action potential, let me just look at your book, because every book has them slightly different. Okay, so the action potential is a temporary change in the membrane potential of the axon. Okay, so it's of the axon, and it actually originates in the axon hillock. Right, so that's that first part of the axon as it branches off the soma, right? And it is a positive 30 millivolts. So if I was to draw my axon again, this where it's negative is resting, right? All of a sudden it becomes positively charged. That's the action potential. So on either side of this, it's resting, right? So I'm going to put this as my action potential, and I'm going to abbreviate that as AP. That is my action potential. So you'll see here that this is temporary. So what's going to happen is it's going to move like a wave down the length of the axon. And this action potential is due to gated channels, right? So for example, these gated channels are important in what is called depolarization. So depolarization is where you go from a -70 millivolts towards 0 millivolts, right? So that means that it's becoming less, the charge is becoming more equally distributed, and that's why it's called depolarization, okay? And this is due to sodium gated channels opening, right? And sodium floods in. Right? It overshoots 0, right? And it goes to a positive 30, right? So it establishes positive 30 millivolts. Okay. So if we look at a diagram that shows this change in voltage over time, it looks something like this. Okay. So here you can see this is resting. And so if this neuron was not being stimulated, it would just stay at negative 70. Right? Here it is moving towards 0, so it's becoming depolarized. It overshoots 0 and comes up to positive 30. But notice how this change is very temporary. So that area that has become depolarized now has to become repolarized. And that is this next part, right? So we have repolarization. Where we go from a negative 30, a positive 30, to a negative 70 millivolts, right? We repolarize. So we're going back to 0, and then we're going way under 0. And this is due to potassium ion channels. So potassium channels open. Sodium channels close. And what happens here is that the potassium being positive floods out of the neuron. And it becomes negatively charged again. Okay. So you'll notice that there's actually, right, this fourth stage right here, which is called hyperpolarization. And this is a temporary thing that happens. That means that the neuron cannot be stimulated again immediately because it has become hyperpolarized. So this is where it overshoots negative 70 millivolts to become more polarized. Okay. And that is also very temporary. Now, it's really important to realize that all action potentials are the same. Okay. So action potentials are all or none. Right? So you can't have a weak action potential and a strong action potential. So for example, the reason why we can tell the difference between a strong or a loud sound versus a soft sound, or a bright light versus a dim light, is due to not the strength of the action potential, but rather it is due to the frequency of the action potentials. So for example, a strong stimulus can actually produce more action potentials over a given period of time. Okay, so action potentials are all the same and they're all or none. Okay. So the part that is important here is, therefore, when does an action potential get generated and when does it not? And so you can see here that there is another component to this graph, which is called the threshold. Okay. So the threshold must be met in order for the action potential to be generated. So that's a negative 55 millivolts. Okay. And so that is what we sometimes refer to as graded potentials. Close this off. Okay. So a graded potential means that there's a change in the membrane potential that does not lead to an action potential. Okay, so a change in charge does not necessarily output. Necessarily. So does not necessarily lead to an action potential. So they're called graded potentials. And so what happens is that a neuron will be stimulated, but if it doesn't reach the threshold, an action potential is not produced. Okay, so I'll put if the threshold is not reached, and that threshold was negative 55 millivolts, an action potential will not be produced. Okay. So sometimes we receive signals and then it doesn't actually get incorporated into our body, into our nervous system, right? So like if the light is too dim, we are unable to see it. Okay. So this shows an example of a graded potential. So notice how it is moving towards negative 50, but it hasn't reached it yet. And so therefore, it just goes right back down without producing an action potential. Sometimes it actually causes it to become more polarized or hyperpolarized. And so a graded potential can actually move it away from the threshold as well. So if we look at some neurotransmitters are said to be inhibitory. And so inhibitory neurotransmitters will actually cause the resting potential to move away from the threshold, whereas excitatory will cause the resting potential to move towards the threshold. And so those are what are referred to as graded potentials. So if it does reach the threshold, right, an action potential will be produced. Okay. So the last thing we'll talk about is how the action potential is propagated. So this is called the propagation of action potential, or AP. This is due to voltage-gated channels. And these cause the action potential to move like a wave. Down the axon. Okay. Now, there are some things that can speed up the propagation. So one thing that speeds up the propagation is the presence of myelin. Okay, so we could put here the presence of myelin speeds up the transmission. And the reason why it speeds up the transmission is it causes the action potential to kind of hop down the length of the axon. So if I'm going to draw my axon here, I'm going to draw a Schwann cell or a oligodendrocyte here and here and here. So I'll say that this is my Schwann cell. Okay. These spaces of bare membrane right here are referred to as nodes of Ranvier. So that's actually named after a person, right? Nodes of Ranvier. So myelin is like insulation. So it's like insulating your copper wire so that you do not lose the charge as it moves down through the wire. And we also want to insulate it like with, if you think about using electrical tape, right? Wrapping it around the cord so you don't get shocked, right? By the electricity moving through it. So this does the same thing. So these are my Schwann cells. And so instead of having to move down the entire length of the axon, it actually hops from one node to another. And only ion channels that are in these bare spaces actually have to open. So the action potential moves, it actually moves down the whole length, but this is insulated. So only ion channels open in the nodes. right? So the nodes have the ion channels. And this type of hopping down the axon is called saltatory conduction. Saltatory conduction. So saltation is hopping. Kangaroos have saltatory locomotion, right? These have saltatory conduction. So does anybody know what neurodegenerative disease is sometimes caused by the immune system destroying the Schwann cells? What is the name of the disease that is caused by the destruction of the Schwann cells? You know? So what does MS stand for? Yes. So this is multiple sclerosis. This is an autoimmune disease. It is not curable, and it is where the immune system attacks Schwann cells.
What are the symptoms for this?
I think pain is one. I think also you start to lose the inability to move properly. So you, in its greater stages, you become bound in a wheelchair because you can't walk. And so you lose control of your motor coordination. But I think pain might be an early symptom of it as well.
And there's no cure? Like
There's no cure.
Like prednisone?
Oh, well, that treats it, but it doesn't cure it.
Okay.
So you can give them steroids because that suppresses the immune system, but it doesn't cure it. So that is just a long-term treatment for it. Now there's actually one that's similar to multiple sclerosis, which is really interesting because it does repair itself, and it's called, I can never spell it right, but I think it's called Guillain-Barré. And this is temporary, whereas multiple sclerosis is not. And it usually follows a viral infection. So multiple sclerosis can have remission, so they can sometimes get a little bit better, but then they generally follow kind of a decline, right? Guillain-Barré is a really interesting one because you'll like be getting over a really bad viral, like maybe the flu, and then you'll wake up and you won't be able to move. And what has happened is that your immune system in fighting off the infection has actually gone too far, and it has demyelinated your axons. And then you end up in the hospital, and it might take you a year or 2 years to get full function back. So with physical therapy, you can recover from the Guillain-Barré, but you can't. So both of these are called demyelinating diseases. So demyelinating diseases, which means that it's caused from the myelin being stripped off the axons. And it's really interesting when you look at children when they're first born, a lot of their axons are not myelinated. And so if you think about babies' movements, like their fine motor control, like they're jerky, right? They're trying to like pick something up and they're just like this, right? And a lot of that has to do with the fact that not all of their neurons have become myelinated. And so after you're born, the motor development you see in babies oftentimes is due to the fact that their axons are becoming myelinated as they develop. Okay, so we'll stop there for today. And then next, we're going to talk about how action potentials are transmitted from one neuron to another across the synapse. Okay, And we'll talk about neurotransmitters as well. Hello, hello.